back. We are live and we are streaming on LinkedIn for another fantastic episode of the Reviewer Credits Ask Us Anything. My name's Gareth Dyke, and as always, I'm joined by my good friend Maria Machado. Maria, how are you today? How's things?
I'm good. How are you?
Very well, very well. Full of the joys of spring as we are streaming live on LinkedIn and ready to talk about the 7 deadly sins of research that can be spotted by peer review. If you're joining us on the LinkedIn feed, you can put any comments, any questions into the comments underneath this LinkedIn event. We'll see them and we will respond. Maria, 7 deadly sins. Why is ethics so important in research and peer review?
So you need to ensure proper use of funding, basically. It comes down to money. People give you money to conduct research that's good for society and for whatever it is that you're researching. So you need to maintain public trust. And to ensure the proper use of funding, you need to promote scientific integrity. Basically, give people proper credit for whatever work they do. So to ensure that you're doing that, to speed up the collaborative process, you need to abide by a certain set of rules. And that is what these types of webinars that we're conducting here are supposed to tell you as well, is that peer review is one of the ways, but not the only one, but one of the main ways that you assure accuracy and accountability in the record of what's published.
We trust research that's been peer reviewed. We use research that's been peer reviewed. We put research that's been peer reviewed back into our own research projects. This aids collaboration. It speeds up the scientific process. And what we're all about at Reviewer Credits is promoting academic research and publishing integrity. And ethics is also really important, as we'll learn in this session today, to make sure that people who've done the work on a project get the proper credit for the project. So ethics matter. And what we do, as Maria has just mentioned, is provide training and support at all stages of the publication cycle. So if you need help understanding the ethics of funding, the ethics of research data, the ethics perhaps of predatory journals, the ethics of research and plagiarism, submission and peer review, which is really what our event today is all about, we're here to help, because peer review, the peer review process, which researchers do without much reward and recognition. That's, until the advent, I would say, of reviewer credits, very important in the academic process. So what we're going to talk about today, and do feel free to jump in with questions and comments at any stage, is about the ways in which we can identify those deadly sins of research using the peer review process. Maria, over to you. 7 deadly sins.
So this was largely based on an editorial that was in Nature a while back, where the 7 deadly sins of research were kind of spotted and identified and separated in between them. So, to go over the 7 deadly sins, most of them have to do with hacking your data to tell a story that you want to tell. But you can do that in different ways. And the different ways that you do that as a researcher may not always be intentional, may not always be something that you're doing, but sometimes it is picked up in peer review, and some peer reviews use terms that you may not even have heard of before. And those ones, I don't know why, they have a lot to do with food. Maybe it's just because I'm hungry and it's lunchtime. They have a lot to do with food. So going them in order of, I think, the least to most severe. The last one is something that we won't even, I don't think that there's lots and lots of examples on it, especially to do with images. There are people that specialize in spotting fabricated images, and graphs and micrographs and histochemistry and all of those things. So there software to do that, and hopefully reviewers won't have to be charged with doing that so much in future. But the ones that you really should look out for, the first one is harking. So how do you hark, which is really something that a lot of people have been accused of at times,
I
which
hadn't
is
noticed that they're all to do with food. I really hadn't noticed that until we do the event. Harking, cherry picking, pea hacking, salami slicing, double dipping, and not publishing negative results. We'll see if you can relate not publishing negative results to food in a little
okay.
bit. Sorry, back to harking. The first of the deadly sins. What is that, Maria? Harking. I've never heard of that
It's
before.
basically hypothesizing after results are known. So in scientific research, you have a hypothesis and then you design an experiment to prove or disprove that hypothesis. But most of the time, what happens is that you've performed the experiment, you've thought of the hypothesis a long time ago, and when you're finally writing up your manuscript, you don't remember what it was anymore, and you try to figure out something that kind of fits with your data. And that's what harking is about, is you come up with a hypothesis after you know of your results. And that just means that spurious correlations, like the number of x XKCD comics published and the robbery rate per 100,000 residents in Vermont, correlate, just because sometimes they do. It's life. It's, nothing, one doesn't have to do with the other. There's no causative relationship there. It's just they happen in tandem.
If you think about it, I think we've all done this in our research careers a little bit,
Yes,
haven't we? I mean,
it's
we've all
very easy
collected
to
data.
do. It's
Yeah.
very easy to do because you do remember kind of what it was that you wanted to start out with, and then you don't anymore because life happens and research goes in a different direction.
And this is the difference between correlation and
And
causation,
causation, yeah.
right? So it's very important as a reviewer and also as a researcher to keep your hypothesis at front of mind when
Mhm.
doing your data collection and then when doing your analysis, so that you don't end up in this situation. Have you seen this as a peer reviewer, Maria?
I've seen people trying desperately to argue that a correlation was causative, and that is bad. So whenever you do this, the problem is not that you've collected data that somehow have correlation with something else, is that you don't claim that one is causing the next.
Interesting, interesting. And you mentioned that we're getting more serious in
Yeah,
terms of ethical violations as we move through our now food themed seven deadly sins. So we're on to number two. This one's definitely food themed, a bit self explanatory, but nevertheless, Maria, cherry picking data. Seen that
yeah,
one?
yes. So whenever you're uh, repeating and repeating and repeating, and sometimes individual experiments, have the same trend. Sometimes they don't, and sometimes you tell yourself, "Oh, I'm not going to include the results of this experiment because it was Easter, or because I was there on a Sunday, or because." No, you should, if you've conducted everything properly, you should include the data that you collected. That has a lot to do with having controls and samples collected at the same time with randomized allocation, so that you're not biasing yourself just because of the way that you're sampling or collecting data.
So it's important that the control and experimental groups are sampled at the same time, and randomization can be used to control for this. I'm not a statistician. I know lots of friendly statisticians. Darko, if you're watching, we
Yeah.
love you. But there are people that you can talk to help you with this kind of thing, to make sure that you're not going to get in trouble or going to get in problems in the peer review process when you could be selectively using data from a bigger sample. I think it's really important.
Yeah, and so this is something that people like to do a lot, especially in biological sciences.
Number three,
Yeah.
not to do with food in this case, although we've tried our best
Well,
with some
kind
cute
of,
little fish.
because it's bee hacking, also known as a fishing expedition. So, what you can do is, if you have something that's on the threshold of significance, of statistical significance, you may want to rethink whether your sample size was enough. You may want to perform more equivalent tests. You may want to collect more data. You may use directional tests to increase statistical power. So these are the times where, for instance, when you're doing a t-test, it's one-sided or double-sided. But most of all, what you should do is, before all of that, submit a registered report so that whenever you say something in your methods like, I'm cleaning up data for analysis, or the parameters were tuned for a specific model, you have basis of saying that that those types of analysis or manipulations or comparisons have a basis and are standardized across your analysis.
So I'm taking my data and I'm scrabbling around within it to look for significant
Yeah,
results, right?
yeah.
The fishing expedition analogy. So just explain a little bit more about registered reports, because I'd not heard of those before this.
So registered reports. There is a whole blog on the Reviewer Credits website to do with registered reports, because that has to do with love methods. So last Love Methods Week, which was the first Love Methods Week, it's where we are talking about how registered reports and protocol sharing, detailed protocol sharing, reviewed detailed protocol sharing, is a very good thing for everyone, because it means that you design your research and it gets reviewed by significance and by study design by a set of reviewers. And then whenever you collect your data and you analyze your data according to the assumptions that you laid out in a separate publication, then it's reviewed by another set of reviewers. So you have much more power in your conclusions.
So it makes sense, much more reproducibility. So I
Yeah,
come up with a method
yeah,
and a
yeah.
technique, and then I publish that as a registered report. That means
Yes.
that when I'm collecting the data, I'm going to stick to the method that I came up before, but there's a published record of
Yeah.
the method. That's a great idea. That's really good. Why don't
And
you
you
do it?
can
It helps, right?
it does help, and you can be accredited by the idea of the method itself. It's really good whenever you have something that you don't know, you're completely in the dark whether it's going to work or not, because at least you have one publication of that project, which, even if it is the method that you came up with. So the next one
Number
is
four. Number
number
four.
4.
Back to the food theme. I can understand this one. I've
Very
definitely done this, by the way, in my research career. Those
salami
good old
slicing,
least publishable
publishable
units, as we used
unit.
to call it.
So the least publishable unit is a very good theory, which is, it's the least amount of data you can possibly encompass in a publication, that it still makes sense to be on its own, right? That it still stands alone as a piece of research that's individual and it progresses the field. The thing is, it is much easier to organize smaller units of data. It's fast to go through the dissemination of information peer review because it's simpler. You can have a more detailed description of each step of what you did, but you do lose the big picture overview, and sometimes it can oversimplify complex phenomena. We're not talking here about the registered reports we just talked about. We're talking about when you have an analysis plan and you came up with, and your complete analysis plan leads you to, I don't know, 20 parameters, and you publish 2 in one paper and 3 in the next and 4 a year from now. It's a bit iffy.
This is easiest to understand in the context of my research, like specimen-based research, paleontology, for example, where you could write a paper about 3 fossils, or you could write 3 papers, each about 1 fossil, and that's
Yeah.
salami slicing. We see it a lot in some fields of chemistry as well, like inorganic chemistry and crystallography, for example. When you're working on an individual molecule within a family of molecules, for example, you might be encouraged. And I remember very distinctly being at a conference in my field years and years ago where a professor from a big US university was encouraging us as PhD students to go for the least publishable unit, as he used to say. And it's easily done. Like, why would you write a paper on a family of molecules when you can publish 6 papers, each on the individual molecules? That's salami slicing. So think about whether it's a good idea for you as a researcher. Surely one big impactful paper in a high impact factor journal is better than 6 lesser impact publications in a series of lower tier journals. I would argue this wastes resources. Certainly that's the case in my field. As Maria mentioned, in more kind of biological, genetics, molecular work, it really leads to flawed meta-analyses. And as we've seen in the examples, like this really distorts the academic reward system. I mean, it's, somebody working on specimens or molecules can just publish more papers. So they get rewarded more. It's very important to keep in mind as a peer reviewer, I would say, right, Maria?
Yeah, because then so this is quite hard to spot when you're peer reviewing, because you never know. If you don't have a registered report, you never know what the ultimate aim is. But for instance, if you're reviewing a paper, like I did recently on organic chemistry, that tells you that we synthesized a compound that was supposed to do this, and this was based on compounds we have previously published, which is fine, right? Because they previously published this, and then now they thought about something else. Could be. But then it says, and now we have one compound that's much better than the other ones at doing this. And next we're going to optimize. So they already know that this is not the final compound. They're still going to optimize it. And then they'll publish another paper saying, no, we've optimized.
Yeah, I mean, think about what's best for you as a researcher. Think about what you're looking at as a peer reviewer. Perhaps our most food-based themed tip, and I think we're on to number 5. I could be wrong. Could be counting wrong. What about double dipping?
So, right. Everyone hates this, right? You're somewhere and everyone has, and you have a little dip. You have guacamole and you have salsa. And people go there with their nachos or their potato chips and they dip their things, and then they take a bite and then they dip the thing again. No, you shouldn't do this. So whenever you're developing a model, you have some data, some pilot data that led you to optimizing an analysis method or a model that you want to carry on then with your research. Whatever you use to develop the pilot is not going to be used in the analysis, because then you're just doing circular analysis. You're analyzing the same thing twice. And that is a very big statistical no-no, because it can lead to apparently significant results being found in noise. And that's what's happened very recently and very publicly with functional MRI data. That's what happens with some bioinformatics epidemiology model fittings. That's what happens whenever you have images that you want to pre-process for some unknown reason. You always have to do something to come up with the methodology, and then whatever data you have, don't use it for your final analysis.
This is really interesting to me because I didn't know about this, because my research is not in this kind of area, but we're talking about developing a model using a data set and then using that same data
developing
set for later experimental analyses, right? Is that right?
developing a model or an analysis method or something that hasn't been done before, that you need raw data to develop to begin with, that you couldn't possibly do without some sort of the data that you later analyze. But you started out as a pilot. You didn't have a power analysis. You didn't have, it wasn't something that you did to then be later analyzed. It can't be glued together with the rest of your data that you then developed with an analysis plan in mind.
And as we mentioned, we're getting more and more serious. So if you see this in a study, or you suspect that this might be happening in a study that you're peer reviewing, this would be something. All of these are things to flag to your editor, the handling editor, but this is a potential reason for article rejection, definitely, right, Maria?
Yeah, because it's giving you false information. It's data that could be spurious, that could be random, and because you're picking it up twice, you're giving it twice the power. So you're analyzing it in a way that's not feasible.
Interesting. Now, my favorite of all of them, let's move on. This one is really topical. Everyone's talking about this all of the time at the moment. If you've got negative results, what should you do with them? Well, you shouldn't not publish them. That can be a problem. It wastes lots of resources.
So, and the peak of what you shouldn't do, this is just before fabrication. This is just before making up data. Whenever you've conducted an experiment and you've analyzed your data and you found no difference, and that's what negative results mean. It's not that you don't have any results, is that there's no difference in what you are trying to compare. Sometimes there's no difference because that's what life is. That's still valuable information. That means that you've conducted the experiment, it's well designed, it's well thought out, it gives you information that is going to be valuable for people advancing the field. They're not going to repeat what you did because you found no difference, and they're probably going to use that information for something different. They're going to advance it. It's always
Absolutely.
very, very important to publish even results that show no difference.
You can do that by preprinting. If you'd like to learn more about preprint servers and how you can do that, like we have lots of content available, like lots of other webinars and training. That's a great way. There's also a number of journals out there. But yeah, researchers, again, like with peer review, we're not incentivized to do this. So that's something that we're working on to try to change, I would say, within the industry, within the academic context. Like there has to be more recognition, both for peer review and also for publications that contain, or preprints that contain, or information online that contains negative results, I would definitely say.
Yeah, and this is one of the big ones because it relates to all of the other ones before, right? So if you're not incentivized to publish negative results, you are more likely to look at your data and go on a fishing expedition. You are more likely to look at your data and look at correlations that have no meaning whatsoever to causative hypothesis, and you are much more likely to maybe let's get rid of this because it's an outlier. No.
Definitely feeling quite hungry at the end of this presentation today, but we're talking our way through the peer review process, really. I mean, these kinds of sins, if you like, could be picked up at different stages of the peer review process, of the kind of pre-flight, article submission, formal editorial selection, that kind of triage stage of the process that we like to talk about. Also the formal peer review component. So, I mean, different sins occur or would be collected or selected or discovered at different stages of the process, I'd say, right, Maria?
Yeah, they're much more likely to be discovered at different stages because of their nature as well. So you need people to look at the publication very, very detailed, in a very detailed way to pick up, for instance, p-hacking. But probably if you don't publish negative results later on, whenever you have a body of evidence and a number of papers being published, you will get people figuring out, oh, this person could have possibly saved me a lot of trouble if they had just published their negative results.
Yeah, and from a researcher's perspective, like, you don't want to do these things, because the worst thing that can happen to you as a researcher, as an academic, is for your work to get published and then for this kind of thing to be noticed later, which could lead to retractions, which could lead to negative responses to your work. And that can be like incredibly damaging to your career, to your reputation, even to your institution's reputation. A retraction as an academic really is the last thing that you want to happen. So not just from the perspective of peer reviewers learning to identify these different deadly sins in the papers and in the articles that we get for review, but also really from a researcher's perspective. And we put together this little summary really of the different, areas where research and publishing ethics touch upon the cycle: safety, submissions, plagiarism, cheating, authorship, which we said we would talk about at the beginning, Maria. We should talk about that now. And also conflicts of interest. What about authorships and recognition for the work that you do?
So there is a very good way of that people have come up with along the way, called credit. It's a taxonomy. It's a way of saying how people contributed to a specific research manuscript, or to a specific piece of research. Not only a manuscript, it can be a preprint, it could be a poster, it can be a presentation. There is, if you look at credit online, there is a taxonomy, there's a website that tells you that people can be authors if they contribute intellectually significantly to the design and of the experiment, if they collect data, if they analyze the data, and if in some way they are part of the process of curating and financing. There are other things that don't qualify as authorship, but can be listed as contributions in the acknowledgements. And it's really important to have that discussion early in the research process, especially if you're a very early career researcher, because, for instance, your position in a string of names on a paper may be significant for your career later on.
First and last, usually in the biological, chemical, and medical sciences. I mean, yeah, really important. Talk about this with your co-authors before you begin the writing process. Agree with your co-authors about authorship order. That's really, really important. We, of course, big proponents of COPE, the Committee on Publishing Ethics, Publication Ethics. Have a look also at the ICMJE.org, the International Committee of Medical Journal Editors, one of the most fantastic acronyms out there in the publishing industry. Yeah, really important to get that right at the beginning of the process, because what I've experienced as an editor, and I'm sure you've seen this as well, Maria, it's happens often. A paper gets accepted and then you get that cheeky little email from the authors saying, we forgot to include X, Y, and Z co-authors. Now, this is very problematic in the context of paid for authorship, which is becoming more and more of a problem around the world. There are numerous dodgy websites that you can go onto where you can buy authorship on an already accepted paper. So obviously, for a publisher, an editor, getting an email like that raises about as many red flags as possible. And I got one just recently. We forgot to add a couple of co-authors to our paper. What happens then?
Well, that's where the credit taxonomy is really good, because then you are going to have to ask them, what was their contribution to the paper? Did they revise the manuscript? Did they contribute to funding? Because getting funding on its own is not sufficient to be an author, and that's what a lot of people don't realize whenever they're very early career researchers, is that you're a head of department just because you got your funding or because you got a grant. If he did nothing else over the realm of your research, design, experiment, writing, it's really complicated to accept that they are an author at that stage.
We have lots of resources on this. Do get in touch with us if you'd like more information. If you're listening to this and you'd like any more information on any of the topics that we've touched on today, it's been very food themed. So I don't know about you, I'm off to get a snack. But Maria, anything else we'd like to add? What are we doing in 2 weeks' time?
Well, we're going to give you an update of our initiative on crowdsourcing peer review resources. So if you want to learn all about learning about peer review, then join us in 2 weeks.
Or indeed, if you'd like to get involved, because this is going to be a great, hopefully comprehensive-ish, as much as we can make it, resource for everybody to use in terms of learning content about the peer review process. Thank you, everybody, for joining us in this Reviewer Credits event. If you haven't signed up yet as a peer reviewer on reviewercredits.com, and I don't know what's going on, because you can get access to accreditation training and really great sets of journals and resources, and you know that those peer reviews that are coming to you are coming from accredited, reputable journals. And what about publishers? Journals, we have more than 1,000 so far signed up on the Reviewer Credits platform, so get involved. Maria, thank you so much. Have a great rest of the day.
Have a great day. Have a great weekend. Bye.